Proteases digest proteins by hydrolyzing the peptide bonds that link together amino acid residues. The red molecule is a small protein. The blue molecule is chymotrypsin, one of a large family of enzymes known as serine proteases. Chymotrypsin's active site is tailored to accommodate amino acid residues with bulky side chains such as phenylalanine residues, as we shall soon see in the red protein. Let's take a closer look at the active site. As we cut away the surface of the enzyme, we approach the active site from the inside. The blue tubular structure represents the backbone of the polypeptide chain. We've only displayed the backbone so that we can see the inside more clearly. Normally, the interior of a protein is completely packed with atoms. The red atoms belong to that part of the substrate protein which fits into the active site. The yellow amino acids are the residues most important to catalysis. Serine at position 195, histidine at position 57, and aspartate at position 102 make up what is often called the catalytic triad. The serine hydroxyl group is the essential catalytic group and is why this family is known as the serine proteases. It will be easier to understand the mechanistic details of the reaction if we simplify the picture, mapping the 3D positions onto a 2D schematic map. Once the substrate is bound, the hydroxyl group of serine 195 nucleophilically attacks the scissile peptide's carbonyl group. The hydroxyl group is made more nucleophilic by the neighboring histidine that extracts the hydroxyl hydrogen, a process that is facilitated by the polarizing effects of aspartate 102. A covalent bond forms between the serine and the substrate to yield a complex known as the tetrahedral intermediate. The tetrahedral intermediate, which resembles the reaction's transition state, is stabilized by two amide hydrogens that hydrogen bond to the anionic oxygen. This region is known as the oxyanion hole because it is occupied by the intermediate's oxyanion group. The tetrahedral intermediate quickly decomposes back to a planar carbonyl group. This can happen in one of two ways. The bond to the serine can dissolve, regenerating the original starting components. This is just the reverse of step 1. Or the bond to the nitrogen can break, releasing the C-terminal portion of the substrate while forming an ester linkage to the N-terminal portion. This latter assembly is called the acyl enzyme intermediate. Note that the hydrogen temporarily held by the histidine has now been passed on to the leaving polypeptide fragment. Next, the ester bond of the acyl enzyme must be broken. This is accomplished by another nucleophilic attack on the carbonyl group, this time by a water that is diffused into the active site. The water transfers one hydrogen to the histidine while forming a covalent bond to the carbonyl carbon. The result is another tetrahedral intermediate stabilized by the amide groups in the oxyanion hole. In the last step, the tetrahedral intermediate decomposes by breaking the bond to the serine hydroxyl group. The hydrogen held by the histidine is transferred to the serine, and the substrate is released with a carboxylic acid terminus. The enzyme is returned to its original state, ready to bind the next protein.